Good evening. Today I will be discussing the effects of pH on Enterobacter cloacae. Before we get started, there are some things to know about me. My name is Dowdy. I'm a senior at Tunstall High School. In the fall, I plan to attend George Mason University, where I will major in psychology with a concentration in cognitive and behavioral neuroscience. One of the reasons I chose this project was in my AP biology class last year, one topic that transferred over from unit to unit was pH, and so I wanted to implement this into my research project somehow. Additionally, Enterobacter cloacae is known to have many adverse effects on many different biological species, including the inhibition of plants' growth, including chili pepper plants, as well as the cause of many different human bacterial infections. Before going into this project, I wanted to understand the role that pH played in bacterial growth. However, this was not one cohesive role, as there are many different classifications of pH. This includes neutral environments as well as extreme. In terms of bacterial growth, neutral environments are anywhere within the range of 6 to 8, and these are the most conducive to bacterial growth. Bacteria has also been found to grow more cohesively in these environments, meaning it grows more in clusters as opposed to spread out over a given area. On the other side of pH are the extreme environments. These are classified as acidic or alkaline. Acidic environments are defined as anywhere within the range of 1 to 5, and alkaline environments are anywhere within the range of 9 to 14. These are the most inhibitory towards bacterial growth. However, there are a few bacteria types that do grow best in these environments, and these are known as either acidophiles or alkaliphiles. While these extreme environments may be used to kickstart certain cellular processes, such as cellular respiration, over long periods of time they are overall unsustainable for life. Learning these things about pH made me want to understand how it played such a significant effect on growth. First, I had to understand that pH is the measure of hydrogen ion potential in a given area. This determines if hydrogen is pumped into or out of the cell. If this is disrupted by introducing a new pH environment to the cell, it will prevent the cell from completing many metabolic processes, including cellular respiration, which is the cell's breathing process. This also makes the cell unable to maintain homeostasis, as it cannot regulate its environment, food production, or movement. Additionally, pH disrupts the ability of proteins, known as antiporters, from properly regulating pH by placing them in an environment too extreme for them to properly pump hydrogen ions into or out of the cell. Similarly to this, proteins within the cell membrane are damaged by pH, resulting in increased permeability, which can allow harmful substances like silver nitrate or excessive proteins to enter the cell. Many plants have antimicrobial abilities which emulate the effects of pH I just discussed. This can be very useful in developing natural treatments to prevent the inhibition of other plants' growth, as well as natural remedies to internal human infections. Some of these natural antimicrobials include phenolic compounds, which have been shown to protect olive leaf plants from inhibition. These phenolic compounds have also been found in plenty within the human intestinal tract, where they have been shown to prevent the growth of harmful bacteria while aiding in the growth of healthy bacteria, showing their ability to aid in the prevention of human infection. These natural antimicrobials can also be found within essential oils from leaves such as the Arcominus plant, which has been shown to inhibit the growth of the Enterobacter species, which is a close relative to the Enterobacter species. While there is much known about pH and Enterobacter separately, my research aims to focus on how these two elements work together. While performing my experiment, my research question was which pH environment best inhibits the growth of Enterobacter cloacae. I hypothesized that if the pH of the environment was 3, the growth of Enterobacter cloacae would be inhibited the most, and that if the pH of the environment was outside the range of 7, there would be no effect on the growth of Enterobacter cloacae. When testing these hypotheses, my independent variable was the pH of the environment, my dependent was the growth of Enterobacter cloacae. I used a control of the pH of 7, which is the pH of pure water, and the nutrient agar I used in my plates, as well as the temperature of incubation, remained constant as to limit lurking variables. I began my experiment by gathering and preparing my materials. This included mixing my chemical solutions for pH and placing my diffusion discs into their respective solutions to soak. I additionally prepared my bacteria and swabs and labeled my agar plates as pictured here. I then randomized my sample by randomly assigning a pH value to each plate. I repeated this process for a sample size of 50. After randomizing my sample, I then began to prepare my plates by evenly swabbing my bacteria onto every plate through the use of a cotton swab, as seen in the image on the right. I then placed my pre-soaked discs into the center of my agar plates, as seen in the image at the bottom. I then placed the plates into an incubator, making sure to place them upside down so that the condensation pictured in the bottom image would not drip onto my bacteria. I then left them to incubate for 48 After incubation, I measured each zone left by the solutions and placed the measurements into an Excel spreadsheet. I then conducted a one-way ANOVA to determine if the inhibition I measured was significant enough to warrant a rejection of the null hypothesis. After performing the one-way ANOVA, the conclusion was drawn that at the significance level of 0.05, I failed to reject my null hypothesis with a p-value of 0.08. This graph depicts the mean growth of Enterobacter cloacae in varying pH solutions, and this can explain visually why my results were not significant. The bars are very close in height, only varying by 1.5 millimeters across the entire graph. The pH of 3 may appear as an outlier in this, which can be explained by the lack of growth data for this group. In the pH of 3, less than 50% of all data points were numbers above 0, whereas all the other groups had data values above 0 for at least 50% of their data. Additionally, there is no growth data for the pH group of 7, as it was used as a control. Because my p-value was above the significance level of 0.05, there was not sufficient evidence to conclude that pH has a significant effect on the growth of Enterobacter cloacae. This also means that the differences in growth between my groups was not significant. Because my results were not statistically significant, I cannot confidently say that there are any significant real-world implications for my research. However, my research does contribute to the field of knowledge, and that other research focused on the strain can concentrate on why it may have had such little reaction to pH. One of the reasons my results lacked significance may be that the discs that I used as my zones of inhibition were not fully saturated with my pH solutions before placing them into the bacteria. Additionally, the concentration of my solutions may have played a role in the inhibition of growth as proposed by a study conducted by Vincehela et al. One of the most glaring limitations of my study was the lack of time. This prevented the ability to complete multiple trials, as well as the time in which I had to review the literature and develop my research question. Additionally, I was limited in resources. The method of measurement, being a large 12-inch ruler on very small petri dishes, limited my ability to be precise. The use of index cards as diffusion discs may have also limited the rate of diffusion and saturation of the pH solutions. Further research based on this topic can explore how Enterobacter cloacae's lack of reaction may contribute to its ability to inhibit plant growth. One of the major things I learned from this project was that success does not always equal results. It's okay to fail. Even though my data was not significant, I still learned the research process. My major takeaway from this project is my love for research. Throughout completing my senior research project, I have fell in love with the research and inquiry process, and this has allowed me to realize that research is what I wish to pursue a career in. This makes me forever grateful for Ms. Long for introducing me to this process and fully supporting me throughout, whether it be helping me find articles, assisting me with my writing process, or creating my agar plates for me. I would also like to thank Ms. Dillon for aiding me in developing my research question, as well as assisting me with handling and incubating my bacteria. A special thank you to Ms. Fain for creating my hydrochloric acid and sodium hydroxide solutions I used to create my pH solutions. Thank you to Ms. Kirby at Tunstall High School for assisting me with the formulas needed to create my pH solutions. And finally, a special thank you to Dr. Bocock and Dr. Mark Jones for approving my research question and allowing this entire process to take place. Thank you. Are there any questions? Yes, Ms. Prost?
What would be the future implications of what you found as far as the actual, in vivo?
Well, because my result, Ms. Prost is wondering the future implications for my research. Because my results were not significant, my future implications were mainly how the lack of reaction may contribute to growth. Can I ask you to elaborate on what you mean by in vivo?
What was the point of doing the research? What were you trying to get at for the future, is what I'm saying.
I was trying to see if pH could influence somehow Enterobacter's growth because of its ability to inhibit plant growth. And so I think because of my results, not there being very little reaction, that lack of reaction may contribute to plant growth.
Okay.
Ms. Davis?
Why Enterobacter?
Ms. Davis is wondering why Enterobacter cloacae. I originally was going to do my experiment on bacteria found on hands, and then I decided to specify a little more, and I researched what bacteria was commonly found on hands, and Enterobacter cloacae was listed as one of the top 5, and it was one of the only ones we had access to as a K-12 school.
How many discs did you put on each petri dish, and did you just use kind of a
how'd you measure the zone of inhibition around each one?
Dr. Brown is wondering how many discs I used per plate. I used 1 disc per plate because we used very small dishes. We had the option of larger dishes, but we chose smaller and just used 1. And sorry, what was
And
your
how did you
second question?
measure the zone of inhibition around
Dr.
each
Brown is
one?
wondering how I measured each zone. I used a 12-inch, well, I took a Sharpie and I marked the edge of the zone of inhibition, and then I measured from the center of the disc and